Folks, I'm very excited today because I have an icon and I have a phenomenal leader and someone I have admired from afar until we met recently. But Dr. Ray Kurzrock, who will introduce herself, but in a little bit to all of you. But to me, she has always represented the progression of precision medicine and molecular understanding of cancer over the years. And I've invited her to the show to talk a little bit about the journey, your career journey, and how your career journey intersected with the progress that we have done with precision medicine. Ray, I really can't thank you enough. I know how busy your schedule is, but I really believe that you are doing listeners a favor by hearing from you and learning from you. So this is I don't say that lightly. So I really appreciate your time that you're planning on spending with me. But let's get started to set the stage by just knowing a little bit about you. Tell us about you, where you are now, but how did you start in the journey of medicine, cancer, and then really pivoting into wanting to work on the molecular underpinnings of malignancies?
Yeah. So first of all, thank you for having me on. And it was wonderful to meet you in Milwaukee and have dinner with you. And just really a privilege to be on this podcast and to be able to speak to you. And again, thank you for the nice comments. I appreciate it. Just a background, I was actually born in Toronto, in Canada, and I came here, after I came here to do my residency in the United States, and then I started to look for a place. I knew I wanted to do research in oncology, and I started to look for a place that was at the forefront of research, and I found MD Anderson. So I joined as a fellow. And I rose through the ranks and eventually became a department chair and founded a new department of early phase trials. At the time, I think they gave me the department because nobody wanted to do phase 1 trials at that point. No, people don't remember this now, but at that point, doing phase 1 trials, they were toxicity trials. Which is something I didn't really believe, but people were still arguing if you could even tell patients that they might expect a response in phase 1. So it's really interesting to me now, the younger physicians, everybody wants to do phase 1. It's really hot. But at that point, it was less exciting, but I actually thought it was exciting, and it was a great opportunity. And we built the department. It became, I believe, the largest department of its type in the world. And it really gave me an opportunity to use some of the things I'd done before I became the department chair to learn from them. So I had been a hematologist. I'd been involved in the chronic myelogenous leukemia field, the BCR-ABL, and then I'd move over to solid tumors. And, people don't even know that I did CML. Or that
I did not know. This is the first time I knew that you did CML.
Yeah. So Aaron Goodman, actually, he was my fellow. Of course, he's very well known now. But he told me one day, he said, "Dr. Kurzrock, I can't believe you did all this stuff in hematology." I actually published the Nature paper that discovered P190 BCR-ABL. So that ages me, but I was only 5 years old at the time. So, but
you worked with Janet Rowley?
No, I worked at MD Anderson, and my own. It was my own lab.
With the CML, did you intersect with Janet Rowley?
Well, I met her several times, right. But of course, she's an icon because of the Philadelphia chromosome. but I didn't work with her directly. But at any rate, so the BCR-ABL story really, was changed everything in hematology. And then at one point I decided I'm going to go into solid tumors, and people said, that's crazy. You are well known in hematology. Why would you make a shift? And I said, because solid tumors is a desert, and we need to learn from the leukemias. So, of course, now nobody even knows that I did leukemias, which is kind of interesting, because they said, like this was a crazy career move. Anyway, because I was familiar with both hematology and with solid tumors, I think I was given the opportunity to be the chair of the phase 1 at MD Anderson. And probably the other reason, because people didn't want it, which is kind of a joke, but probably somewhat true. But anyway, it grew to be a very large department. And this was the era when we first started to have drugs that were targeted. And some of those drugs, interestingly enough, were not being, even though they were targeted, they weren't being developed in to be treating patients who had the target. So we implemented a protocol in our department. It's actually 2 protocols, one called Predict and one called Impact, which matched patients with the targeted drugs. And these were phase 1 patients. They were solid tumor patients with metastatic disease that were refractory to therapy. And we began to be able to see responses that we never would have believed could occur. So it was still the early days, but a lot of the targeted agents that we use now routinely were actually developed during that period of time. And during that period of time, both because of the experience with BCR-ABL in the leukemias, but also because we began to see responses in the solid tumors, I came to believe that cancer was entirely a genomic disease, and that it was, so this is a little far out, but I'm going to say it anyways, that the classification of cancer by organ of origin was an accident of history. And it was because the light microscope, which is used to classify cancer, by looking at the surface of the cell, and then you can tell, oh, it comes from the colon, or it comes from the lung, or it comes from the breast, was invented 400 years ago. And the molecular microscope, which is next generation sequencing, only came to be in the year 2001. And of course, at that point, it cost $3 billion to sequence a patient. So because sequencing is so new, we don't classify patients by their genomic sequence. We classify them by their organ of origin, because the light microscope is 400 years old. But the driver of cancers is not the organ of origin, I believe. It is actually the genomics. I think the tissue is, I'm not going to say irrelevant, but minimally relevant compared to the organ of origin.
So let's back up just a little bit. When you were started to build a department, and then you were tasked by that, how do you even start? Like, what do you do? I mean, you're starting there. Do they give you funds and they say, go do whatever you want? How do you even start? What's your first couple of things that you did to start building something that became an empire, frankly? But how did you start?
So I think it's kind of an interesting story, and I think it's maybe even unique to MD Anderson and the personalities that were there. At that time, San Antonio, which is quite close to MD Anderson, really had the best phase 1 in the world. And MD Anderson had a lot of patients that were going to San Antonio. And Ki Hong, who was my boss, he kind of didn't like that. MD Anderson has a lot of pride. So he decided that we needed to have a phase 1 department. And then he decided that I would be chair of that department. And this is the way Ki Hong was. He gave me a mission. He said, build me the best phase 1 in the world. And, you know, I mean, it wasn't more detailed than that. So, you know, I started to hire people and so forth. And I've always been kind of conservative in the way that I use money. But here I was spending a lot of money, and I thought, you know, hiring a lot of people, and we were bringing in studies. So I thought the money would come in, but I wasn't sure, you know, I'm kind of running a couple of $1 million behind already. Not sure if the money's going to come in. So I went to Key and I told him, in retrospect, this is kind of amazing to me. So I told Key, you know, I'm spending all this money and I think it's going to come in, but I don't know. You know, I could be $2 million behind. And Key looked at me and he goes, what did I tell you? And I said, what are you talking about? He says, I told you to build me the best phase 1 in the world. And if you do, as long as you're doing that, I will cover you.
Wow.
You're covered completely. You know, I didn't realize how special that was. So, you know, only Key and I were in the room. I never even dreamed to say, Key, can you put that in writing? It didn't even occur to me to say that. Key, can I have like something in writing about what the package is? I absolutely knew that what he said in that room that nobody else heard, that his word was good.
Isn't that amazing?
And yeah. So, and I don't think, I know now, having, you know, grown more, that was amazing. It didn't strike me as amazing then, but it was amazing. So probably, you know, I do have some innate ability to operationalize and build, but I really want to give Key a lot of credit because I felt safe, because I felt that as long as we were doing what he asked us to do, which was make the best phase 1, that if I ran into any problems, and it didn't matter how much the money was, he was going to cover me. Now, the truth was that all the money came in and I never had to ask him for anything. But the confidence that I had, that his word was good, I think was really important.
I mean, that's really what leadership and vision are. Ray, so obviously you knew from the CML work that you've done that there's cancer could be molecularly driven, but like you said, in the solid tumor world, it was a drought. What was the event that happened in your career on the solid tumor front as you were building the phase 1 at MD Anderson that was an aha moment where you said, okay, everything I thought about in solid tumor, it's actually indeed true. I can find that thing. And, you know, in our research medical career, there's this event that happens that really you always think about as made a difference. You mentioned one with Kwan, who said, just go do it. Don't worry about it. That stuck with you for decades. Take me through the medical piece.
Yeah, so I don't think it was one event, but it was several events, but I'll give you one of the events that I won't forget. So I had a patient with medullary thyroid cancer. He was very advanced and he was pretty near death, and he had a Ret mutation. And so we began to think that maybe we should use a Ret inhibitor. And sorafenib was already available. And we knew from its profile, it wasn't marketed as a Ret inhibitor, but we knew it was a Ret inhibitor, but we couldn't get a hold of it. So I started looking at our portfolio of drugs and calling the companies and saying, is your drug a Ret inhibitor? Because, you know, a lot of these small molecule inhibitors do more than one thing. And one of the drugs we had was a drug called XL184, which is now cabozantinib. And but at that time it was XL184. And I called the company. It was brought into clinical trials as a MET inhibitor, not Ret inhibitor. I called the company and I asked them, is it a Ret inhibitor? And the medical director looked at the list of targets and told me it's not on the list. Okay. So then it occurred to me, I called back the next day because I was getting kind of desperate. And I said, is it not on the list because you didn't test it, or is it not on the list because it's negative? And so she looked again. She said, well, it's not on the list because I didn't test it. And so I said, well, can you test it? And she said, well, that's hard to do. And I said, no, it's not hard to do. I told you that earlier that I had found the P190 BCR-ABL, and the way we found it is we did kinase assays in our laboratory. They're actually pretty simple assays. It's, you know, you have the answer in a few hours. So I said, no, it's not hard to do. I know you know it's easy to do because we do kinase assays in our laboratory. You'll have the answer in 24 hours. So anyway, she went back and she looked at it and she says, a very potent Ret inhibitor. They put so we put the patient on the clinical trial, and he had a phenomenal response. I mean he probably had 3-4 weeks left to live, and he had a response that lasted more than 4 years, but his response was immediate. So this was, you know, in a patient, just indicated to me that what we had seen in the leukemias, we could see in the solid tumors as well. It wasn't the only example, but it was an example that I can't forget.
And that's the, you know, I mean, the description or the story that you just shared with us is probably what's now, it's an example of what people call precision medicine. And there's, you know, there are people who hate that term because they will always say, well, we've always done precision medicine. If you have a sore throat and I give a Z-Pak, it's precision medicine. I treated you with the right therapy at the right time with the right drug. But that's really simplifying what truly precision medicine is. When do you think that term precision medicine as we know it today, like the molecular understanding of cancer, really became what it is today? I can't think of when we started calling this precision medicine.
Yeah, so I think that I do think your sore throat is precision medicine, because in infectious diseases, we culture the organism and then we find the right antibiotic. Of course, as sometimes we know that usually a sore throat is caused by this or that. But if we're not sure, or if we have an infection that is an important infection, we're also always going to culture the organism, and we're going to give an antibiotic that matches it. So it is precision medicine. In cancer, it was originally called personalized medicine. And then it evolved to be called precision medicine. But I think both names are actually accurate because what we now know is that the genomic landscape of patients is complicated. And therefore, in order to be precise, we have to individualize the therapy or personalize the therapy. So I think in a way, both names are correct. The precision name, because we want to be precise, and the personalized name, because we want to individualize therapy. But the precision name, I think, has really stuck. And I think it came around probably about 2010 to 2012 that really became popular to call it precision medicine.
And, I mean, as you developed the program, you said it was called phase 1. And then and correct me if I'm wrong, I'm just trying to remember the history. It became Department of Experimental Therapeutics as opposed to phase 1, or still had the phase 1 name?
No, it was initially phase 1 and then became Department of Investigational Cancer Therapeutics. But the emphasis was on early phase trials and eventually on tumor agnostic trials. I think we probably ran the first tumor agnostic trials in the department. Around, I'd say 2007, 2008, we put a protocol through for a tumor agnostic trial of temsirolimus, which is an mTOR inhibitor for patients that have had PI3 kinase, AKT, mTOR pathway abnormalities. And I don't know for sure, but I think that was probably first ever tissue agnostic trial. The IRB didn't like it much, but eventually they did approve it.
Is there a story behind changing the name from phase 1 to investigational? Is there a story there why it changed in terms of the department name?
Not a very exciting story. I think it turned from what was a program to an official department, and I don't think I don't think the name, like, that was what Key wanted. He wanted that name, so I think you can hear what Key wanted, Key got.
Yeah, so then you mentioned I'd like to talk about the first tissue agnostic one because look, I mean, in my opinion, and the opinion in many, you were and are visionary in how you really thought approach to cancer is. But I have to predict or guess, and maybe I'm wrong, that you probably faced some resistance early on. If I take you to 20 years back when you were trying to sell the idea or explain your vision of how this is. And it's funny that you mentioned the temsirolimus study that the IRB were not, they were not too excited about. So take me through some of the challenges you faced as you're trying to convince people that this is a reasonable way to investigate cancer, whether it's colleagues, administrators, IRB, pharma companies. I mean, you basically were selling your vision, and I presume there were not a lot of buyers back then.
No, I don't think there were a lot of buyers. And I think the story that I remember the best, I was reminded of it because I went into my garage a few months ago. And I moved to San Diego and from Houston, but you find things in your garage and they're memories, right? So I had almost forgotten about this, but now I remember. So I think it was about 2008. The NCI asked for transformative grants, okay, like transformative ideas. So I had a transformative idea, the molecular reclassification of cancer, which meant that cancer was a genomic disease, it was tissue agnostic. And I want to tell you that I've had a lot of NCI grants, and I think I write very well. I'm not going to, I've had a lot of grants. I publish a lot. I know how to write. So I don't think that was the problem. So I put in this transformative idea, the molecular reclassification of cancer. And I forgot about it. I probably put it out of my mind. It got the worst possible score that you can get. So at that time, it was scored from 100 to 500. And I think they gave me a 499, which was a message. They did not like this idea. So I think that reflects the kind of resistance that was there to that idea that cancer was a genomic disease and that it would eventually be reclassified. I think that resistance is still there, but it's a lot less than it was around 2008. Let's put it that way.
Do you think the resistance today is from the funders, from the researchers, or from the patients or the oncologists who are treating patients? Because there are many oncologists out there that would say, well, great. I mean, you can tell me the genomic features of the cancer. It's not going to really affect how I'm going to treat the patient. I mean, so you gave me a lot of information, thank you, but I'm still going to do carbotaxol.
Yeah, so I think that the patients understand it the best, in my experience. When I talk to a patient, they totally get it. I mean, and they don't have to be educated to get it. They really understand it. I think that there's resistance from all other factions, and some of it is the way we've structured everything. And that's actually the hardest thing to get around. So, we've structured our practices, our clinical trials, our academic departments around diseases. You know, you're a GI doctor, or you're a CNS doctor, or you're a breast doctor, or you're a lung doctor. And now to come along and say, well, genomics is the driver. How do you restructure that? It's not easy to actually restructure that. And by the way, I'm not saying that histology has no effect. I do think that histology adds some context, although that context may be because of secondary genomic abnormalities that are common in that histology. In other words, I'm not sure that it's the histology that actually gives it context, or the secondary genomic abnormalities that might be found there. But I have no doubt that the primary driver is the genomics. But it's really hard to restructure everything we built in the last 50 or 100 years of doing cancer into a new paradigm. I think that's actually some of the hardest part of it, is how do you restructure if this is a genomic disease? So I think it's much better than it was a decade ago as far as accepting genomics and the importance of genomics, and not just genomics. We're getting into transcriptomics and immunomics and proteomics and so forth. But genomics is at the head. It was the first, it was the first out. So I think the resistance is now much, much less. But we're still not completely accepting that this is the fundamentally important issue, is what are the genomic aberrations in a cancer?
You did the first tissue agnostic study now 15 years ago, something like that. That's a long time ago for the first tissue agnostic study. And today we're taping this at the end of 2022. It's going to air early 2023. We have several drugs that are approved by the FDA for tissue agnostic indications.
Right.
How did you convince the IRB with the first study? And if you speculate in the future, do you see more tissue agnostic indications happening?
Yeah, so that's a two-part question. I'm not actually sure how we eventually convinced the IRB. I think it was just persistence. And one of the things I used to teach my young faculty was when you get no, that's not the end of the negotiation. It's only the beginning of the negotiation. So I can't tell you how many young faculty came in my office with clinical trials or anything else, and almost in tears, or just very upset, and said, it's a no, and I'm going, no, it's not a no. That's the beginning. Well, that's the beginning of your negotiation. We're going to go back again. So I think some of it is a matter of persistence. And I think it took us a long time to convince the IRB, but we just kept on going back and, trying again, and probably took us about a year to get the trial activated. Now, I think there's going to be a lot more tissue agnostic approvals. I can't speak for the FDA.
What is the most, like, the folks who don't believe in the tissue agnostic indications? What do you hear? What's the opposing view of this? Do you hear like folks who say, well, this isn't like what? The folks who disagree with this approach of tissue agnostic, what do they contend?
So what they contend, the most important thing I think they contend, is that in an individual histology, you don't know that this works. Like, how can you extrapolate to a histology? And then they always use colorectal. It's such a great example. BRAF inhibitors and colorectal don't work. Now, of course, they do work, but you have to give them with the EGFR inhibitor. So encorafenib is approved, but you have to give it with cetuximab. The argument is, so how do we know it works in this histology, and how do we know it works in this histology? And one of my passions is rare cancers. So there's 186 rare cancers. And so my answer is you really cannot do the study in all 186 rare cancers. So the contention that I hear most often is, how can you approve something when you haven't tried it in that rare cancer? And my counter argument is, I mean, we can't do a study that looks at 250 cancer types. It's just not possible. So that's where the argument is. And let me see if I can articulate why I think that argument is incorrect. So the argument of the opposition that I've heard is, what if we make a mistake and it doesn't work in a certain rare cancer? And my counter to that is, I don't think that's going to be true, but I'm going to give you that argument, and I'm going to agree that we are going to make some mistakes, even though I may not even believe that, but I'm going to allow you to win on that argument that we are going to make some mistakes. But that's not really the question. The question is, do we lose more lives by occasionally making a mistake because there's some exception to the rule? Or do we lose more lives by having to wait years to test every single histology? So even if the opposition is correct that and I don't actually think they're correct, but I'm going to give it to them that they're correct, we're going to make some mistakes. My contention is that those mistakes will sacrifice fewer lives than waiting for years before we give an approval by having to test hundreds of histologies. I mean, literally, if you want to test hundreds of histologies for each target, it might take you 100 years. Think about the number of lives you would have lost by waiting. So I'm not sure I've articulated that well.
No. I think the point is really very well taken, and I certainly got the point you're making across. But this is a very nice segue into another thing that always comes up when it comes to precision medicine. With the idea is that, if you have a targeted therapy, you may think it is the right thing to do, which is good, but you really must compare it in a prospective randomized controlled trial against current standard of care, which could be whatever chemotherapy du jour that we are actually doing. For the most part, these targeted therapies go through an accelerated approval by the FDA, and they go through like single arm trials and all of this. And there are obviously two camps. One camp is you really must do a randomized controlled trial, and the other camp is we really can't do it. And, I mean, I can tell you, I'll go on the air and I'll say that if I have a mutation and there is an actual drug against that mutation, please do not randomize me and give me the drug that goes against the mutation. I do not want to be randomized. So I'll say that. I say that publicly. But I mean, you're a scientist and you hear these things. So how do you convince others who say you really must do this? Or take me through. Imagine you are on the podium in a debate. Which we sometimes like to do it in scientific meetings, and take me through the talking points that you try to articulate to the opposing view.
So it's the same talking point that I tried to articulate before. We know that randomized controlled trials are the gold standard, and they're the most likely to give you the right answer. By the way, they're not perfect either. With a p-value of 0.05, if you run 20 randomized controlled trials, just by chance alone, one of them may be positive just by chance alone. So randomized controlled trials aren't perfect. But that's not the argument. We are going to agree that they're going to give you the best answer. And they're going to give you the least likelihood of approving something that is, of not approving something that is actually inactive. Okay, so we're going to completely agree with that. And we're going to also tell you that if we continue to proceed with accelerated approvals, which historically have not been really rescinded, they've turned out to be correct because the FDA has put a bar, at least in oncology, of very high response rates. But I'm going to agree that the chance of making a mistake is higher with an accelerated approval than it is if you have a randomized control trial. So we're going to concede that. And then I'm going to say once again, but that doesn't tell you which way sacrifices more lives. And so I'm going to argue that because randomized controlled trials are so much more difficult to run, take so much longer, especially in rare subsets, that it will take years to answer the questions by randomized controlled trials. And in those years, in trying to avoid a mistake, we will lose many lives because the drug is not, or the drugs are not available for patients. And that if we continue to proceed with accelerated approvals, yes, it's possible that we will eventually have a mistake. And the likelihood of a mistake is higher than with the randomized controlled trials. But nevertheless, the number of lives saved will be greater with the accelerated approvals than if we wait for the perfect answer. Because waiting for the perfect answer means that we're waiting years, and all those patients, you have to consider the patients that won't get the good drugs, along with the minority that might get a mistaken drug. So overall, I'm not arguing that randomized controlled trials aren't the best trials. And I'm not arguing that we're not going to make mistakes. In fact, I'm conceding we're going to make mistakes. And I'm conceding that randomized controlled trials are the best trials. But I'm arguing that more lives will be saved by the accelerated approvals than for waiting for the perfect answer from a randomized controlled trials. During that waiting period, thousands and thousands of people will lose their lives because a good drug was not available. So again, I don't know if I've articulated that clearly enough, but that is my counter argument.
But yeah, you did. But I just want to, maybe I'll ask just a pinpointed question, because I think you've made a couple of points that are really important. But I'm going to just theoretically, let's go back to your medullary thyroid cancer patient that you mentioned, who has again a Ret Fusion, and you have a Ret inhibitor that is out there. I mean, is the right way to do this methodically and academically is to have a randomized controlled trial where patients who have known Ret Fusion will be randomized to a Ret inhibitor versus any standard chemotherapy? It just sounds to me that I realize that this appears the most pure academic approach, but I would struggle consenting a patient to that.
So you bring up a very good argument that I didn't make, but is a very correct argument, and that's the ethical argument. Do you feel comfortable randomizing a patient to the wrong therapy? One of the basis of randomized controlled trials is equipoise, meaning that you don't actually know, or you think you don't know, which arm is better. So And if there's a randomized controlled trial, that means that you don't know 100% for sure which arm is better. But if you have a really strong feeling based on science and observation that one arm is better, how do you randomize that patient to the wrong arm? And I think that's why you don't see these randomized trials in precision medicine. If you have an NTRK fusion and the response rates are someplace between 50 to 80%, and the response rates to chemotherapy are pretty negligible, how do you randomize a patient to the wrong therapy? And then you mentioned this yourself. Would you agree to be randomized to the wrong therapy? Never. And I don't think as oncologists, most of the oncologists that I know that treat patients, like we really want our patients to get better. We have altogether too many patients that die. And giving patients what we feel is the wrong therapy, I don't think oncologists are comfortable with that. And that's why they haven't done these randomized trials.
I think you hit the nail on the head with a couple of things, but it's worthwhile clarifying to listeners. I mean, there's always the argument that it's not necessarily the wrong therapy. It's just the newer therapy may not be as good or better. I mean, in other words, what we're saying, sure, you can give chemotherapy to this patient, and you may think that the target therapy is better. Doesn't mean this is wrong. It just, this may not be more effective. But I think the equipoise that you mentioned is really, really, really hits the nail on the head. Because if you have evidence or belief based on science or observation that this treatment is likely going to help your patient more, you can't really then just put them in a different therapy. So that's at least that's how I feel, and I'm glad that you feel the same.
Yes, and I'll add one more thing. The better the therapy, the harder it is to do a randomized trial. So we wrote about this in one of our papers. What is the cutoff? Like, how good is a therapy before you can't do a randomized control trial? I mean, we used an example, which is at the extreme. Could you have ever done a randomized control trial in type 1 diabetics of insulin against whatever, pick your you can't, because all patients without insulin would die. Most drugs in oncology are not 100% like that. So it's not as clear as insulin. But I think the example of insulin, people can understand it, because with 100% response rate versus 0, you can understand that you wouldn't want to do a randomized controlled trial. So now the question is just, what is your cutoff? Is it 50% versus 20%? I don't know exactly the right answer, but I do know that a lot of these targeted therapies have high enough response rates and durable enough responses. And the traditional therapy that the patient might get have low enough response rates that most oncologists wouldn't be comfortable. And I don't think patients properly informed would want to go on the placebo arm or the control arm.
Yeah. So in order to understand the molecular profiling of tumors, obviously, you need to sequence the tissues and all of this. And without going into detail, I hear a lot that, when you do these sequencing, you're going to see so many things. And for the most part, what you find is not going to be helpful. I mean, you might find a target that has no drug, or you might have a drug that is not approved for that target, for example, for that particular indication. Or you might find a lot of targets, or you might find, one target that has a drug. So why did you really discover all of these other targets, all of this? In your teaching to fellows and junior faculty, what do you tell them about how to approach this?
Yeah.
And has your vision about this shifted over the past 20 years?
Yeah, so I think we are asking some very important questions. So the first thing is that we're finding targets more and more often, and the poster child is non-small cell lung cancer. We are today about 70% of non-small cell lung cancer have some sort of actionable target, important actionable target. Each of those targets, I mean, there's a lot of them. There's BRAF and EGFR and ALK and on and on. Each of those targets are in a small subgroup of patients, but when you add all the subgroups together, it makes up 70% of patients. So we're not into now rare anymore. Now we have enough targets that substantial numbers of patients have a target. I would argue, however, that even if it's rare, like NTRK, which is 1 in 300 patients, I don't know when in medicine we said, well, that's rare, so we're not going to figure that out. Because for that patient that has it, it's extremely important. So I definitely think every patient should be sequenced. The other thing we know is that there may be certain targets that are more common in certain diseases, but there are many tumors that refuse to read the book. And they have a target that they're not supposed to have. One of the targets I read about and I first found on Twitter, it was from a paper I think was published in JCOPO, but I became aware of it on Med Twitter, was a glioblastoma, which you know is a very deadly tumor that had BCR-ABL and was treated with imatinib. Somebody tweeted about it and I went back, I think it's JCOPO. So the books say that BCR-ABL is found in CML, it's found in a subset of ALL, and it's found in a smaller subset of AML. Well, this glioblastoma didn't read the book, and it had BCR-ABL. And so if the physicians had not done sequencing, they would not have known about that, and they wouldn't have been able to treat the patients. Unfortunately, many tumors don't read the books. And so there are a lot of exceptions to the rules.
I like that when you put it, the tumors don't read the book. It's perfect. There's a book in your story, Ray. I really, just listening to you, and I'm not joking. I mean, just explaining, I mean, there's a lot written about this, but I think there's a different perspective, in my opinion, for folks who took care of patients and did the science as well, and really saw the evolution of these and really changing over the years from resistance to acceptance, all of this. Have you thought about chronicling your journey and writing something?
I don't know, baby. I've had a few people that have mentioned it, but it's a thought. I think it's the journey of cancer. So it's interesting how it's evolved. I do want to mention what I think is the next level, and I think it's very important. And you alluded to it earlier, and it's that many tumors have multiple alterations. And what do you do with that? Especially in the metastatic tumor setting, most tumors don't have one alteration. They have more than one alteration. And when I was at UCSD, when next generation sequencing came about, we were so excited because before we were doing PCR on a few genes and had a few hotspots. And then we got next generation sequencing. I'd moved to head the personalized medicine program at UCSD, and this was like end of 2012. And as we began to sequence patients, we were very excited to have this data. And then we found out that we had a problem, and that was all this data and all the alterations that were present in a patient. So we started a paper with my colleagues. We started a protocol with my colleagues, Jason Siklik and Shumai Kato, that we activated at the beginning of 2015. It was called iPredict. And what took us 2 years to get it through the IRB. We started in 2013 to write it, and it was approved in 2015. But the idea was to give customized combinations that match each patient's molecular portfolio. Nobody had ever really done that before. So every patient, we looked at their molecular portfolio. And in this case, we used Foundation Medicine, but it doesn't really matter. Foundation Medicine, Tempus, Caris, whatever sequencing you use, if you do next generation sequencing, you know that there's multiple alterations. So we tried to maximize the number of alterations that we matched. And we've now published a whole series of papers on our experience. The first paper was in Nature Medicine in 2019. And the bottom line is that we found that we could give customized combinations safely by adjusting the dosing. And the second thing that we found, which is very important, was the greater the degree of matching of the drugs to the molecular alterations, the better the outcome. Every outcome parameter was better. Response rate, progression-free survival, overall survival. And what do I mean by greater degree of matching? I'm going to give you a simplified version. If a patient has one alteration and you target that one alteration, you would get a score of 100%. But if a patient has 10 deleterious alterations and you target one, you get a score of 10%. If the patient has 10 deleterious alterations and you target 7, you get a score of 70%. So the more alterations you could co-target, the better the patient did in every single parameter. So this is how I treat patients in our precision medicine clinic now. We try to co-target the maximum number of alterations by giving customized N of 1 combination therapies to patients.
Ray, when you find those two mutations and you want to do two drugs together, do you go back and do like a phase 1 3+3 to decide, or how do you decide on the dose? Just how do you do that?
So I learned a lot from this. Remember, I'm a phase 1 doctor, so I should believe in phase 1 trials, but I think phase 1 trials need to be revamped. So think about how you get dosing from phase 1. And basically, on 6 patients, if more than one of 6 have a toxicity, now you've got the maximum tolerated dose. Of course, you expand that a little bit, but basically you end up with the same dose for your 20-year-old Olympic athlete who unfortunately got cancer and your 85-year-old fragile woman in a wheelchair. They end up with pretty much the same dose. Now, just think about that. Doesn't make any sense. So what we did is when we do a new combination that hasn't been in the literature, we reduce the dose. And I'll give you some rules of thumb, although it's a little bit more complex. If we put 2 drugs together, we reduce the dose by 50%. If we put 3 drugs together, we give a third of the dose. We see the patient every week, and we titrate the dose to tolerance. And we discovered a couple of things from that. One thing we discovered is that you don't have to give the FDA-approved dose. You can give way lower than the FDA-approved dose, and you can have efficacy. So that's number 1. And number 2 is I now believe it's actually a better method because we're not just personalizing the drugs, we're personalizing the dose. So that 20-year-old may end up on full dose everything. And the 85-year-old may end up on compromised doses, which they should have ended up anyways with lower doses. And we showed through the trial, it's in the publication, that we were able to do this very safely. I actually think now it's safer to do intra-patient dose escalation and figure out the right dose for every patient than taking a dose that's off the shelf and giving everybody the same dose.
Amazing. It's hard to believe we've been talking for almost an hour, but I don't get bored talking to you. So maybe in the just last question is, how do you spend your day right now between patients, research, lecturing, teaching, mentoring, and you know, how do you spend your time and divide your time, and what do you do for fun?
Fun? What's fun? First of all, Um, I would say I'm in clinic about 1 to 2 days per week, and I have a precision medicine and rare cancer clinic at Medical College of Wisconsin. I have an interesting commute relationship because I still
Yeah.
live in San Diego, and I have some young faculty that help me. I serve as a consultant role, but I'm very excited about the precision medicine and rare cancer clinic. I also have a wet lab. I have a dry lab, bioinformatics, and I'm always trying to teach. And I love the science, and honestly, I love the patients. And I think, to me, it's very integral to who I am, to be able to try and bring new therapies to patients.
You have this
So
annual conference. What annual conference that you do? You were in Europe recently with the WIN conference, no?
yeah, the Worldwide Innovative Network for Precision Medicine. This was started by John Mendelsohn when he was president of MD Anderson. And the mission of the organization, which I love, is to bring precision medicine globally. So the conference before COVID was every year in Paris. Of course, we took a break during COVID, and this year the conference was in Barcelona. And fun.
You picked some nice, not too shabby cities to have this conference. I gotta get myself to this conference, I guess, next time. When is it? Where is it going to be next year?
So we're not sure yet. So we used to have the conference every June in Paris, and honestly, it's a wonderful conference. I mean, it's really good. It brings together people from everywhere, and actually, we should bring you there. Absolutely, I think
Well,
you're perfect for it. And my husband and I would take like 12 days. We'd go to Paris for the conference, and then we'd spend a few extra days in Paris. Then COVID came along. We didn't have it for really 2019 was the last year we had it until now. And we had been thinking about changing to Barcelona. And this year we had it in Barcelona, and it was in June. So we haven't decided yet when it's going to be next year, but I think there's an adjustment in the post-COVID era to, should we do it every year? What city should we do it in? But I will tell you, we weren't sure because we hadn't had a conference since 2019 if it would work. But it was fantastic. We had attendees from all parts of the world, and the talks, a lot of talks put everybody to sleep. These talks were phenomenal. No, these talks were absolutely phenomenal. There was great audience participation. I think WIN, has really done an excellent job with putting together the conference.
Well, Ray, thank you so much. This has been a delightful hour, and really appreciate you spending some time sharing some nuggets with us and just your journey. I can definitely attest that a lot of folks look up to you, and just you really have transformed a lot of the way we approach cancer. And there are many people that their lives have been impacted positively by your work, even though you did not care for them directly, because this work that you have done is leading to a lot of approved therapies that really make people live longer. So thank you.
So thank you. And again, it's been such a privilege to talk to you and really to meet you and everything that you're doing for this field. I really
Yeah,
admire what
I'm just a host of a podcast. That's all. You're the one who's doing
you're
the work.
you're much more than that. And again, pleasure to talk to you. And I hope we meet again in person soon.
Absolutely.